Here is the neuromuscular junction. The presynaptic cell is a motor neuron. The portion of the muscle cell here with the motor end plate. Motor end plate is the part of the cell membrane of the muscle fiber that meets the somatic motor neuron. Here we have nicotinic cholinergic receptors. Acetylcholine released by the mechanism that we have already discussed. Just to recap, action potential in the somatic motor neuron. This is where you're looking at the saltatory conduction that is skipping these myelin sheaths. And this is the synaptic knob. And here you're talking about calcium channels, voltage-gated calcium channels being open in calcium ion, entering the axon terminal and moving the vesicle towards the cell membrane and ultimately releasing the neurotransmitter into the synaptic cleft. And now the binding of the neurotransmitter, acetylcholine, to the acetylcholine receptors. And now the ion channels open. Here the ion channels yet again release both sodium and potassium ions, however, in opposite directions. More sodium ions make their way in compared to the potassium ions leaving the cell. Okay, so these are referred to as chemically gated cation channel. We could also call it as ionotropic channel, ligand-gated channel, all those names that you're familiar with already. Okay, so the same thing is shown in a step-by-step manner. In here, we'll run through this quickly. Action potential at the NMJ from a somatic motor neuron, opening of voltage-gated calcium channels, release of acetylcholine reaching the motor end plate, binding to post-junctional acetylcholine nicotinic receptor. This is asked over and over in your board exams. That's why I have highlighted this. Ligand-gated channels open. These are on the postsynaptic cell, nicotinic receptor, and we talked about them as ionotropic receptors, greater sodium ion to enter motor end plate, then normal depolarization. Depolarization in the muscle fiber is called end plate potential, or EPP. Basically, it's depolarization. Before we called it as excitatory postsynaptic potential, or EPSP. Now we call it as EPP. Okay, which will become action potential. Please note, summation is not needed here. The channels are large and multiple channels are open simultaneously. So there is depolarization that automatically leads to action potential. Summation is not it. So now, interestingly though, it travels in both directions. Remember, in the neuron, it would travel in one direction only, but here in the muscle fiber, it can go in either direction, both directions. It actually spreads in both directions. And actually, this is sarcolemma, down the sarcolemma, not the sarcomere. So, and through the T tubules, sarcolemma first and then the T tubules, okay, muscle contraction. I'll change this to sarcolemma. Okay, acetylcholine is degraded by acetylcholinesterase. This is a classic step. Acetylcholine is released. The next thing you notice is it is being broken down by its enzyme acetylcholine esterase, okay, to prevent continued prolonged muscle contraction. There are disease conditions when acetylcholine esterase is not produced in enough amounts and not remove the acetylcholine that's released, that triggers continuous contraction by the muscle fiber, resulting in spastic paralysis.